Mitosis is the growth and development of cells, as well as repairing damaged cells and replacing old ones. Mitosis starts with interphase, where 90% of cell life is spent. There are three components to interphase: G1, rapid growth of the cell; synthesis, where chromosomes duplicate; and G2, which consists of additional growth. In interphase, the nuclear envelope in the nuclear membrane contains chromatin, which is 50% DNA and 50% protein. As well, there's a nucleolus, and the asters are together at one pole. In the next phase, prophase, the nuclear envelope disintegrates, the nucleolus disappears, and centrioles move to opposite poles. The chromosomes shorten and thicken, and spindle fibers form. In metaphase, chromosomes line up single file on the equatorial plate, and spindle fibers begin to reach out to the chromosome's centromeres. In anaphase, spindle fibers attach to the centromeres of chromosomes and pull the sister chromatids apart, pulling them to opposite poles. In telophase, the nuclear envelope reappears along with the nucleolus, and the chromosomes thin out. In cytokinesis, the cytoplasm splits. In an animal cell, the cleavage furrow pinches off, and in a plant cell, the cell plate develops. The result is two diploid daughter cells. Meiosis is the process of undergoing spermatogenesis and oogenesis for sexual reproduction. Meiosis is a combination of two parts. Meiosis 1 is a reduction division starting with interphase. In interphase, the same rapid growth, G1, synthesis, and additional growth, G2, occurs. In prophase 1, synapsis occurs, in which the homologous pairs find each other and form a tetrad. Crossing over can also occur, increasing genetic diversity. The nuclear envelope and the nucleolus disappear, and centrioles migrate to opposite poles, and spindle fibers form. Metaphase 1, homologous pairs line up randomly on the equatorial plate, also increasing genetic diversity. In anaphase 1, homologous pairs segregate from each other and move to opposite poles. In telophase 1, the nuclear envelope and nucleolus reappear, and the spindle fibers disappear. In cytokinesis, the cleavage furrow pinches off, and the result is two haploid cells. The two haploid cells from meiosis 1 continue on into meiosis 2. In prophase 2, the nuclear envelope disappears, centrioles migrate to the opposite poles, and there is no crossing over and no synapsis because there aren't any homologous pairs. In metaphase 2, double-stranded chromosomes line up single file along the equatorial plate in random assortment, increasing genetic diversity. In anaphase 2, sister chromatids are pulled apart at the centromere. In telophase 2, the nuclear envelope and nucleolus reappear and spindle fibers disappear. In cytokinesis, the cleavage furrow pinches off, and the two haploid cells from meiosis 1 undergo meiosis 2. The result is four genetically unique haploid cells.